It's a pleasure to be here in Brazil and to share the work of a very large team with you today. I have intellectual property and patents in the field of cell and gene therapy. That conflict is managed in accordance with University of Pennsylvania policy and oversight. I want to start off with bridging an idea from a lab to a clinical trial. And we start with the idea, knowing the introduction that you have just had, that tumors have evolved mechanisms of immune evasion and immune suppression that include decreased immunogenicity, active tumor-induced immune suppression, and antigenic modulation. In other words, CD19 or whatever target we choose, there may be a cell that does not express that antigen or that epitope. So how do we fix this? And that is through design. So there was already some background on B-cell malignancies, but I'll point out some prognostic implications of the presence of lymphocytes. And this is work done by Luis Parada at the Mayo Clinic, looking post-stem cell transplant in lymphoma, the predictive value of a high lymphocyte count. And you can see it is quite distinct for a lymphocyte count greater than 500. We also know from other data in ovarian cancer that the presence of tumor-infiltrating lymphocytes, or TILs, is also predictive of progression-free survival and survival. So there is then this rationale for effector T-cell therapy, that following chemotherapy and stem cell transplants, T-cell counts and immune function are depressed for months to years. And if we think of tumors and viral infections in the context of evolutionary means, our hypothesis then is that we can select, re-educate, and expand T cells ex vivo outside of the tumor microenvironment and reinfuse to prevent relapse or infections. So if we could design T cells for the treatment of disease, they would be potent, they would have good memory, they would be persistent, and they would be numerous. So when I started working on T cells and the mid-90s, there was thinking on the left that you would repeatedly stimulate cells either with viral antigen or with tumor antigen, and then after 6 weeks you would have a large number of cells to reinfuse. And there were mixed results with this approach. We know now that this tends to exhaust the T cells, and they have a limited ability to persist long term. So our idea was to perform a polyclonal stimulation to maintain the entire repertoire and then rely on stimulation within the body. But we now know that we can redirect these polyclonal T cells by gene transfer. So how do we build T cells? And thinking about the merging of biology with engineering, what I'm going to share with you now is how we generate genetically engineered T cells or polyclonal T cells, even in the laboratory. I had the opportunity yesterday to see the nice facility that you have built here, and where you have the opportunity to engineer these cells. So I'm going to start, let me just back up to the previous slide and go through the general idea. And the general idea is that we need a source of cells from blood, from bone marrow, from tumor. If we're taking tumor-infiltrating lymphocytes, we have a step then to perform washing, enrichment, and purification. We then have a step to perform activation of the T cells, and we can insert a gene at that point, a period of ex vivo culture to expand the cells necessary that we need for a clinical dose. Then a step for washing and removing the residuals, and final product formulation. And this final product is the drug that is infused into patients. So here I've listed some various equipment that we can use at these different steps. There are some others that I haven't listed, but I'll walk you through a typical process where we generate engineered T cells. So we start with the collection, and the collection is on equipment that is used to collect stem cells for stem cell transplants. These are pheresis machines. We have both in our facility, and we're able to collect for every 1 liter of blood that is processed through the machine, 1 times 10 to the 9th, or 1 billion white cells. Now, healthy adults have a blood volume of 5 liters, so we can collect in first pass 5 billion. We normally process through the machine 10 to 12 liters, so we may have 10 billion white cells collected, and the red cells and the platelets are returned to the circulation. The next step to enrich for lymphocytes is shown here, and this looks like an older version of a pheresis machine, but it actually is a modified rotor that allows the separation of lymphocytes and monocytes. And there is a separation chamber here, a closed system disposable kit, and the way that this instrument works is shown here. If you imagine centrifugal force going from left to right, what you do is pump buffer in the opposite direction from right to left. And in this way, if you increase the speed of the buffer flow in stepwise fashion, you can separate the red cells and the platelets, and then the lymphocytes, and then the monocytes and the granulocytes. So in this way, you can have monocytes to generate dendritic cell vaccines, and you can have lymphocytes to generate T cell products. So now let's go to the next steps, the activation. Shown here in the right-hand side, colored in yellow, are beads that are a composite of polycarbonate plastic and iron with surface chemistry that allows for covalent linkage of proteins. We link on the surface of these beads antibodies directed against CD3 and directed against CD28 that deliver 2 signals necessary to provide full activation of T cells in the absence of providing a negative signal through CTLA-4. We also have a cell-based artificial antigen presenting cell. This is a K562 cell line. It's actually a leukemia cell line. And we can insert genes in these cells, high affinity Fc receptor, and we can then decorate antibodies on these high affinity Fc receptors as well as ligands such as 4-1BB-L. Now, whether we add the beads or the cells to T cells, we can stimulate T cells to divide substantially in an ex vivo system, and this is a log scale. If we add those beads every 2 weeks, we can get a 12 log 10 expansion over 2.5 months. That is equivalent to approximately 40 population doublings and shows you the capacity that your T cells have to divide in a suboptimal ex vivo system. So we design our clinical cell processing so that we harvest the cells at day 9 or 10, so they have all this residual capacity to divide in the body and to find tumor and to kill it. We need a way to deliver genes, and we use a viral vector as a Trojan horse to deliver the gene. In our case, we're using a disabled form of HIV that efficiently transduces primary T cells and enables stable long-term expression. We culture the cells initially in gas-permeable bags. It's very efficient when we're working with large volumes, and we have the opportunity to maintain a closed system. We then transition to a bioreactor, shown here. And in this case, the bag is not gas permeable, but gas is pumped into the bag, 5% carbon dioxide. And this platform shown here rocks back and forth. and then you get a wave motion with the media, which is why the original company that developed this was called Wave Biotech. That facilitates much more efficient gas exchange, and you can grow cells much more concentrated than you can in a static situation. You can also perfuse in fresh media, and there's a filter sitting on top that keeps the cells in the bag while you remove the waste media and insert fresh media. And here's a picture showing the control module. You can vary the rocking rate and the angle and the perfusion parameters. And this shows the whole setup. Now, biotech and pharma use this system to generate proteins and antibodies. This is a commercial version of 500 liters. It's the size of a bed. Now, we don't use that to generate T cells because we're working on autologous products, but it shows you that this technology has spread throughout biotech and pharma, I believe throughout the world, to generate therapeutic proteins and antibodies. At the end of the expansion culture, we removed the beads that we used to stimulate the T cells with the magnet. Shown here is one version. Here's the version that is currently commercially available. This is sold by Thermo Fisher. And now at the end, we formulate in bags and cryopreserve or gene-modified products. Products that are not gene-modified can be infused fresh at the bedside. But before we do the formulation, we wash, and we actually use intraoperative blood transfusion equipment that we have repurposed to wash white cells instead of whole blood. And they operate on the same principle. You can collect cells into a reservoir. There's a spinning bowl. You can wash the cells, remove waste, and then formulate. And this version from Haemonetics, you can adjust the pump speed and the centrifuge speed. So in adapting this piece of equipment, we needed to demonstrate to the FDA that we had adequate removal of residuals, and we did that by spiking in fetal calf serum and showing that we had a 10,000-fold reduction of bovine transferrin. Okay, so now let's go to CAR T cells and remind you of our genetic redirection. The naturally occurring T cell receptor must recognize antigen in the context of the major histocompatibility complex. And we can insert T cell receptors that are redirected to tumor antigens, but these are limited by tissue type. In North America, HLA-A2 is the most prevalent, but only 40% of the population has HLA-A2. The next common is 20% of the population. The next common is between 5 and 10%. So you can see that if you're trying to cover 100% of the population, you need many different types of T cell receptors that recognize the antigen in the context of different MHC. Now, the chimeric antigen receptor is not restricted by MHC, but it does recognize only surface antigen, which is approximately 1/3 of the total antigen in a repertoire for cancer antigens. But a chimeric antigen receptor is off the shelf. It is composed of an antibody recognition domain on the outside of the cell and T cell signaling domains on the inside of the cell. And now you can think when you insert this, you can redirect those polyclonal T cells to see any cancer antigen to which you can generate an antibody. So now we combine the method of T cell stimulation, the viral vector, the lentiviral vector, and that technology. The lentiviral vector comes in, carries double-stranded RNA, uncoats, reverse transcription to DNA, permanently integrates that DNA into the genome of the T cell. That is expressed on the surface of the T cell, which also expresses its native T cell receptor. So now these T cells are bispecific, but the chimeric antigen receptor is going to recognize the tumor antigen and kill the T cells. So it was in the late 1980s, 1987 by a Japanese group, in 1989 by an Israeli group, that showed that you could construct these chimeric antigen receptors. And this was at the time of discovery of the B-cell antigen receptor, or antibodies, and the T-cell receptor. And the groups had the idea, well, what if we learn more about the function of these receptors by swapping out the pieces? And then you can make the logical leap to redirecting immune function by inserting chimeric antigen receptors. So it was approximately 10 years from these publications that the first clinical trial was conducted. This was proposed by a company called Cellgenesis that presented to the Recombinant DNA Advisory Committee in 1994 and initiated their clinical trials in 1997. And the results from those trials in HIV were published in 2000. This was not an antibody on the outside, but actually native CD4 that binds to GP120 on HIV-infected cells. This group also, this company also conducted the first clinical trial in cancer targeting TAG72 in colon cancer, and those results were only published about a year ago. Now, we knew of this work by Cellgenesis. We, in fact, had been collaborating with them using the bees to stimulate T cells, and wanted to start trials in cancer at the University of Pennsylvania, where we began looking at the design of different chimeric receptors with different T cell signaling domains. And shown here is looking at 4-1BB and CD28, and showing that the 4-1BB had a very powerful effect in terms of CD19 ALL in this mouse model. Well, when we manufacture living drugs from people, and as varied as we are in this room, patients with disease are even more variable, and we get different source material that we collect by leukapheresis. Especially in CLL, we have low T cell counts, low T cell percentages, and the manufacturing is quite challenging. In some patients with lymphoma, we have high monocyte percentages, and the manufacturing is quite challenging. We can use the elutriator to remove the monocytes. We can use the CD328 beads to positively select, using a magnet, the T cells. So while we do have some manufacturing failures at the very lower limits and then the upper limits of monocytes, by conditional manufacturing pathways, we can improve our success in generating the T cell products. And that's shown in cartoon fashion here with variable incoming products. We're able to generate a consistent T cell product. So now I've showed you the background, and you've heard the background on B-cell leukemia and lymphoma. I've showed you the manufacturing. Well, what does that really translate to? And to be able to translate, we have built a team, as you are building here, to enable translation from the research laboratories, scaling up to do the manufacturing, to conduct the clinical trials, and to do the analysis. So we have basic research labs, we have process development and scale-up, we have validation, the manufacturing, the quality control testing on the cell products, as well as regulatory support and correlative studies, as well as the operations and management. And that is all within the Center for Cellular Immunotherapies. You saw this slide in a different version, so I won't go through it, just to remind you that CD19 has wide coverage, even wider than CD20. And this is another version of the manufacturing, so I won't go through that in detail, just to remind you that we are combining the chimeric antigen receptor technology, the bead stimulation, and the lentiviral vector transduction. So the first 3 patients that we treated in 2010 had advanced chronic lymphoid leukemia. These are patients that had failed all available therapies and were not eligible for a stem cell transplant. They enrolled on our clinical trial in 2010. This is patient number 1. 67% of the purple dots that you see are leukemia, and this is in his bone marrow. 31 days later, following infusion of his own engineered CAR T cells, less than 0.5% of the purple dots that you see are leukemia. So a complete remission induced here where he had been resistant. Here's patient number 2, refractory to Rituxan and bendamustine, received his CAR T cells. You can see the dramatic effect on his leukemia, and he was an ongoing partial responder out to 8 months. And patient number 3 was a manufacturing failure. We had only 300 million total cells, 5% transduction with the CAR. He received 1.4 times 10 to the 7th CAR T cells, a dose we would give a mouse. And yet, with his nodular disease, 31 days later, you can see decreased. He was a complete responder. These 3 patients were reported in 2011, and I'm pleased to tell you that patient 1 and patient 3 originally reported as complete responders then, more than 7 years later, are free of leukemia and leading healthy lives. And when we calculate the disease burden in all compartments, the blood, the bone marrow, the secondary lymphoid tissues, between 1.3 and 3.5 kilograms of leukemia was killed by these CAR T cells in the several weeks after infusion. And that's using a conversion of 1 times 10 to the 12th tumor cells equals a kilogram. By the most sensitive assay we have, high throughput sequencing, we analyzed in patient 1 and patient 3 and several 100,000 cell equivalents. We have no leukemia reads where 99.7% or 90% in this patient was leukemia. Again, in peripheral blood and bone marrow, and this shows the time point at 6 months. To give you some ideas of the differing kinetics in these patients, here is another patient showing in this gait 66% leukemia. We're looking for the presence of the CAR T cells here. At day 6, this goes down to 3%, and at day 8 to 0.5%, we start to see the expansion of these CAR T cells. Remember, this is a living drug. The T cells see their target, they can divide, it's a force multiplier. They can then go on to kill other tumor cells. And then less than 0.1% by day 10, both in peripheral blood and in the bone marrow. Here's a patient that at month 2 has no evidence of disease in the blood and the bone marrow. This is a patient that had failed 10 prior therapies, had transformed CLL, 17p deletion, ibrutinib resistant, and radiation therapy resistant. Now the solid lesion here is decreased at month 2, but you can see it's still present. It continues to decrease over several months, and this reflects some different challenges with solid lesions. One is geometry, only accessible from the outside. The second, we believe, is the immunosuppressive environment here in this lesion and perhaps also vascular access. Nevertheless, this patient was a complete responder. Now, thinking about pharmacokinetics and drug dosing, what is the dose of a drug that is dividing? Is it what you infuse or what it is after it divides? And what we observed in the larger cohort of CLL patients is that the complete responders are shown here on the top row. you can see robust peak and long-term engraftment. The partial responders somewhat less, and the non-responders even less. Now, we can calculate the area under the curve and show that it is the area under the curve that correlates with the clinical response and not the dose that is infused. Shown here is the different ways that we're measuring engraftment, both by flow cytometry that is shown in the circles and by PCR that is shown in the triangles, and these do directly correlate. And this is long-term engraftment showing out to 4 years in these first two patients. I can tell you that in these patients, 6.5 years later, we can still detect their CAR T cells. And again, using this sensitive assay, no leukemia detected at 3.5 and at 4 years. So I showed you the leukemia goes away, the CAR T cells are present, but how do we know that there is the correlation between the CAR T cells and the leukemia? Well, here is function shown by removing blood from the patients and then taking the T cells and mixing the T cells with CD19 expressing targets. And if you compare the top row here to the bottom row here, we can see the specific secretion of MIP-1 alpha, MIP-1 beta, and CD107A that is a marker of degranulation. So these functional correlates demonstrate conclusively a mechanism of action for the CAR T cells acting against CD19 positive targets, even 870 days after the infusion of 1 dose of CAR T cells. So a durable response here. Now I want to talk about one particular patient out of that initial cohort of CLL patients, and that is patient 10, and show you that what is remarkable is that one CAR T cell can eradicate CLL. This is a patient that initially had a lower engraftment and then had a peak that came at 50 days. And when we looked at the T cell receptor V beta families, this peak at day 50 was composed of one V beta family, V beta 5.1. One can look at day 28 after infusion and see a polyclonal repertoire. At day 51, you can see 98% of that repertoire is one clone. We can look another way and see the evolution of this clone post-infusion in the blood. And by inference, show that this one clone kinetically emerged and peaked at day 50, which corresponded with the disappearance of a large solid tumor lesion in this patient that up until day 50 was a non-responder and then converted to a complete responder. So how do you know that you have the right cell, which is why we're infusing a larger number of cells. But coming back to the number of cells infused, you can see no statistical correlation here between clinical response and the dose of cells that is infused. Now, in a very large number of patients, there is a statistical correlation, but you can see the substantial overlap. So we are not making a cutoff on cell dose to say that we know that that will be predictive. But we do know that the quality of the T cells is what is predictive. So up until this point, I've shown you clinical data in CLL, and then the question was, well, CLL is a slow-growing disease. What would happen in a rapidly growing malignancy? And to answer that question, we opened up a clinical trial at Children's Hospital of Philadelphia. And to remind you of the survival curves in second relapse ALL, it is dismal. The first patient enrolled at Children's Hospital was a 7-year-old girl who had been diagnosed in May 2010 and had relapsed twice, came to Children's Hospital, received high-dose Cytoxan and clofarabine that would kill an adult, and only 1/3 of children make it through. She did make it through, but still her bone marrow was packed with 60% leukemic blast, and she had lesions in her kidney, liver, and spleen, and she was not far from her demise. She received her CAR T cells in mid-April 2012, this time with no additional chemotherapy. And here's what happened in her. This is looking for the CAR T cells by an anti-idiotypic antibody. This is all leukemia. This is 6 days after infusion. 23 days later, we see the massive expansion of these CAR T cells in the bone marrow, and the leukemia is gone. So a deep remission induced by her own engineered cells. Well, that is the good news. The bad news is that these T cells are very active. They are secreting chemical messengers that mediate an inflammatory cascade. And in Emily, we saw levels of IL-6 that peaked at 10 times higher than we had seen in any adult. She was intubated for breathing difficulties and on 3 different medications to maintain her blood pressure. And she was so sick that her physicians told the family to call everyone in because she wasn't going to make it through the night. Well, this data was being transmitted to Carl June and to Stephen Grupp, the PI at Children's Hospital. And Carl June's daughter has juvenile rheumatoid arthritis. And he knew that there was an approved drug for juvenile rheumatoid arthritis called tocilizumab that happens to block the IL-6 receptor. He saw this data, communicated with Steve Grupp to check the Children's Hospital pharmacy if they had tocilizumab and to give it to her. They did. She received the drug, and within hours her breathing started improving, and she was weaned off the medications, her breathing tube removed, and ultimately discharged from the intensive care unit. And this syndrome, the cytokine release syndrome, was refractory to steroids, refractory to Enbrel, which blocks TNF, but we have now learned from Emily to be able to integrate the use of tocilizumab in the management of the cytokine release syndrome in children and also in adults. We also observed trafficking in not only in the blood, but in the cerebral spinal fluid. And this is important because in a subset of patients, this is a mechanism for leukemia to escape the, and spread. But we do see CAR T cells shown here in the cerebral spinal fluid. This is in CSF, and this is in blood, and we know from the frequency of the CAR T cells at this time point that you're likely looking at what a CAR T cell looks like here. So the same high throughput sequencing assay was used in Emily, and in 1 million cell equivalents, no leukemia. But I'll also point out at the day 23 time point that there are some very few reads here. So what this shows is a 5 log reduction in tumor with no chemotherapy prior to the CAR T cell infusion, and that it took 6 months for all tumor to become unmeasurable, which points to the advantage of long-lasting CAR T cells. So here is Emily in mid-April 2012. You can see the effects of her disease. She's having a popsicle because the cells are cryopreserved in DMSO, which has a garlic taste and odor, and probably 7-year-olds with leukemia are allowed to have popsicles whenever they want. I'm next going to show you a picture of Emily from May of this year. 5 years after her treatment. And I can show you all the data, but nothing speaks to how well she's doing than this picture. And she's meeting famous people. So her parents have started a foundation to benefit pediatric cancer research and support our work. And I'll tell the same story I told yesterday, which is her father, Tom, got a call from the White House that said the president would like Emily and you and her mother, Carrie, to be present, but you cannot tell anyone where you are going. So they took her out of school. They went down to Washington, to the White House, and they arrived and they were ushered into a separate room before the event to meet the president. He comes in. Her father, Tom, shakes Obama's hand and says, "Mr. President, I'd like to introduce you to my daughter, Emily. She had leukemia and she was treated with her own engineered T cells." And Obama says, "Stop, Tom. I know all about Emily." And he gets down on his knee to shake her hand and says, "Emily, how are you doing today? What can I do for you?" And Emily says, "Well, none of my teachers know that I'm here. I really could use a note for school." So he got out his pad, he wrote this note: "Please excuse Emily from school. She was with me." So I'm told the school did accept this, and actually it doesn't have a date on it, so I don't know if she used it again. But she's all good, and she's doing very well in school. She's a very good writer. But Emily was the first, but certainly not the last. We have now treated approximately 150 children in Novartis, about an equal number. This is showing the breakdown by disease, and at 1 month, in minimal disease, there is a 100% complete response rate. In moderate level disease, greater than 5% blast, and 88% complete response rate. And in very high disease, and this is a population that is not responsive to blinatumomab, an 82% complete response rate. Now, there are some relapses. They're shown in red due to two mechanisms. Early disappearance of the CAR T cells. Remember the lower engraftment in that CLL cohort? To solve that, we can give an additional infusion of CAR T cells to keep that area under the curve higher. The second mechanism is disappearance of the CD19 epitope. There's a frame shift, and that leaves those leukemia cells invisible to the CAR T cells. The solution to that is to have another CAR in the garage, and we have a CAR targeted against CD22 that is now in clinical trial, as well as other groups also targeting CD22. 2/3 of the children that came into the Children's Hospital clinical trial had already failed a stem cell transplant, some of them more than one. Even in this cohort, we have an 89% complete response rate. We know that severe cytokine release syndrome correlates with high disease burden. So that gives a hint on to be ready to administer the tocilizumab as the antidote. But so here is the correlation with IL-6 levels and interferon gamma levels. And we're interested to know, can we predict early by disease burden, but in a more finite way, looking at markers of inflammation or early after infusion to be able to administer tocilizumab early. And to answer that question, while IL-6 correlates, it does not predict the severe cytokine release syndrome. So an analysis that was done in collaboration with Children's Hospital, David Tichy, and Pam Shaw and Simon Lacey, Joost Mellenhorst published this paper in Cancer Discovery a year ago, showing that in the full cohort of adults and children, SGP-130, interferon gamma, and IL-1 receptor alpha 3 days after infusion, you can confidently predict the patients that will get severe cytokine release syndrome. And in the children, it's a combination of interferon gamma, IL-13, and MIT1 alpha. So now we have a clinical trial that we opened with 2 cohorts. There is a high disease burden cohort that will get early tocilizumab, and then the regular cohort that gets tocilizumab with the standard algorithm that we developed. So, reminding you where we came from, this is the dismal outcome of second relapse ALL. We signed an alliance with Novartis in 2012 to develop this technology to transfer it to them so that they could perform their own manufacturing and their own global clinical trials, which they have done. So I'm showing you the results of that global trial, which was named the Eliana trial. And this overall survival curve directly overlaps with what was observed in the single-center Children's Hospital clinical trial. A 6-month overall survival of 89%, overall remission rate within 3 months at 82%, and this is the data that was submitted to the FDA in February of this year that resulted in approval, and that was preceded by a meeting of the external FDA advisory committee in July. So this clinical trial was conducted at 27 sites in 10 countries on 4 continents. And I'm going to show you here the lymphoma trial. So moving from leukemia to lymphoma, we also did conduct a trial in non-Hodgkin's lymphoma at Penn in 3 histologies, diffuse large B-cell lymphoma, follicular lymphoma, and mantle cell lymphoma. The Novartis trial, the Juliet trial, was conducted in diffuse large B-cell lymphoma. And again, this duration of response, 79% relapse-free at 6 months, overlaps with the Penn Single Center clinical trial. So the meeting of the FDA Advisory Committee on the pediatric leukemia trial was in July. Novartis spoke, Children's Hospital, Stephen Grupp spoke, the FDA spoke, and patient families spoke. And at the vote, it was unanimous to recommend approval. This was a 10-0 vote. This is Emily's Instagram page. She was there. Her father, Tom, spoke to the FDA. And on August 30th, we received word of approval by the FDA. What was CART19 in Philadelphia became Tisagen, Laclusil, and Kymriah. And I was in Boston at a meeting scheduled to give a talk on CAR T cells in the afternoon. I'm in the morning in the back row checking my email. One comes up from Novartis with subject line, "Congratulations." I open it. It says, "Today, Kymriah was approved by the FDA." And there have been planned at Penn a T-shirt flash mob to celebrate. This is hundreds of colleagues gathered in the Cancer Center to hear our Cancer Center director and Carl June speak, celebrating the approval. Imagine being amongst hundreds of your colleagues for this celebration, but you're in Boston having to look at the pictures. So I never thought as a PhD I would have the opportunity to participate in the development of an FDA-approved drug, but here is the label. It is Kymriah. It's a bag of cells, but it is a drug. And we are conducting at Penn many clinical trials targeting CD19 in leukemia, in lymphoma, also in myeloma targeting BCMA, and in solid cancers targeting mesothelin and mesothelioma, pancreatic and ovarian cancer, targeting c-Met in breast cancer and in melanoma, targeting EGFRV3 and glioblastoma, and most recently targeting PSMA in prostate cancer. We've treated, this number is outdated, we've treated now about 340 CD19 patients and about 100 other CARs. So we have a platform now where we can swap out that antibody domain and target different tumor antigens. And what I've shown you is a synthetic biology toolbox where we can modify these chimeric antigen receptors with second receptors that may recognize another antigen like CD22 or maybe a tissue antigen. We can construct logic-gated CARs that will operate in an and or not fashion, checkpoint-resistant CARs, CARs with safety switches or suicide mechanisms, or conditional or stealth CARs that will only be active if we add a small molecule. And this is a review that we published last year that goes through some of these permutations. So a truck will secrete an anti-tumor cytokine. A universal CAR knocks out the major histocompatibility complex. A self-driving CAR expresses a chemokine receptor. Armored CAR knocks out the checkpoint receptor. A self-destruct CAR adds a small molecule to remove the CAR, and then a conditional CAR is only active when you add that small molecule. A marked CAR can select the transduced cells, and then here are the logic-gated and or not CARs that either in the same molecule or different molecules can bring together and activate the signaling or deactivate the signaling. So this is the Center for Advanced Cell Therapy at the University of Pennsylvania. We have suites for doing cell manufacturing. We have what may be the best view of any clinical cell production facility that I know of, but we have also a very talented team of people that will grow these cells. So some lessons learned in conclusion in developing first-in-human clinical trials is that cells from healthy humans are not the same as cells from patients with advanced disease. We have to do our preclinical work also on cells from cancer patients. And to demand your research laboratories beginning with that material and with clinical grade materials and methods early. That currently variable cell material requires human judgment and intervention. More human intervention is opportunity for deviations, which requires more training. Few very well studied patients can radically accelerate clinical development. And in thinking about water access, we have hundreds of components that go into the manufacture, from the plasmids to the vector to the media to the plastics to the reagents. We have to focus on reducing the cost of goods and services and the labor, increasing consistency, comparability, managing challenging cases, and looking at implementing rapid release test development, and recruiting, training, retaining our skilled technologists and engineers. So in the near term, we do have a limit on the trained staff, and there is an ethical question on the allocation of who is able to enroll, who is able to receive the approved drug, like the first days of the approval of AZT in HIV, and imatinib in CML, and now ZMapp and other Ebola vaccines and treatments. So in the near term, we are outscaling, not upscaling, because we're not going to a 5,000 liter batch that will treat hundreds of patients. It's one lot for one patient. So I'm a representative of a large team that makes these trials happen. I recognize Carl June, who's director of the Center for Cellular Immunotherapy. Our initial philanthropic support, our support from Novartis, and especially the participation of the study participants. I want to very quickly mention the International Society for Cellular Therapy, of which I'm a member. And as you look to develop your programs, participating in activities and meetings with the ISCT that has membership from academia, from industry, and regulatory professionals, to have everyone speaking together is important to advance the field. There is the annual meeting in Montreal, and I hope some of you will be able to participate in that or in local South America region meetings. So these are our patients, and the Whitehead Foundation had a gala event where they invited children that had received CAR T-cell therapy. This, on October 28th, was the largest gathering ever of CAR T-cell patients, 24 children that had received this therapy, from 4 years old and 5 years old and 6 years old. And this is Barbara Netter, the philanthropist that initially funded our work in 2004. So to see her amongst these children was extraordinarily touching. So with that, I will stop and thank you for your attention, and happy to take any questions.